In this lesson, we will be taking a closer look on bacteria, their structure, and their characteristics. As you have previously learned, the origin of this microorganisms extends back to at least 2.9 billion years ago. With such long evolutionary history, we can say that bacteria have amazing skills when it comes to adaptability. For our class objectives, we will understand the nature of bacteria, classify bacteria based on cell morphology, review differential staining procedures, and identify selected important groups of bacteria and associated diseases. We have learned that organisms in the domain bacteria are broadly divided into three phenotypic categories or categories based on their physical characteristics. Those that are gram-negative and have a cell wall, those that are gram-positive and have a cell wall, and those that lack a cell wall. But with a compound-like microscope, we can further examine the size, shape, and morphologic arrangement of various bacteria. Bacteria come in many different shapes, but the vast majority are one of these three general shapes. First, if the cell is spherical or ball-shaped, the bacterium is described as a coccus. Cocci may be seen singly or in pairs, or diplococci chains, or streptococci in clusters as staphylococci in packets of 4 or tetrads, or packets of 8 or octads. Examples of medically important cocci are Enterococcus, Neisseria, Staphylococcus, and Streptococcus. Here are some examples of commonly known cocci and the diseases associated with each type. Note that each of the species of bacteria already gives you a hint on the arrangement or morphology of each one. Example, Streptococcus pyogenes is a streptococci. Next, a cell that is cylindrical is termed a rod or bacillus. They may be short or long, thick or thin, and pointed, or with curved or blunt ends. They may occur singly, as in pairs or diplobacilli, in chains or streptobacilli, and in long filaments or branch. Some rods are quite short, resembling an elongated cocci, as they are called cocobacilli. Examples of medically important bacilli are Escherichia, Klebsiella, Salmonella, Pseudomonas, Bacillus, and Clostridium. The third general shape of bacterial cells is curved. If it is gently curved, it is a vibrio. Curved bacteria usually occurs singly, but some species may form pairs, as in a gull wing. Another spiral cell is the spirochete, a more flexible form that resembles a spring. Different species of spirochetes vary in size, length, rigidity, and the number and amplitude of their coils. Examples of medically important curved bacilli and spirochetes are Vibrio, Campylobacter, and Borrelia. Because bacterial cells look two-dimensional and flat with traditional staining and microscope techniques, they are best seen with the scanning electron microscope, which emphasizes their striking three-dimensional forms, such as in Figure 4-20. Some bacteria may lose their characteristic shape because adverse growth conditions, like the presence of certain antibiotics, prevent the production of normal cell walls. They are referred to as cell wall deficient bacteria or L forms. Some CWD bacteria revert to their original shape when placed in favorable growth conditions, whereas others do not. It is also somewhat common for cells of a single species to vary in shape and size. This phenomenon is called pleomorphism. An example of cell wall deficient bacteria is Mycoplasma. Because they have no cell walls, mycoplasmas are resistant to antibiotics that inhibit cell wall synthesis. Different staining method remains the universal basis for bacterial classification and identification. These staining reactions involve an attraction of the cell to a charged dye. Let's take a look back on the staining techniques that you have learned. Staining techniques are used to observe bacterial cell morphology. A simple stain is sufficient to determine bacterial shape and morphological arrangement. It uses methylene blue as its one and only stain. We also have two types of differential stain. First, the gram stain, which has become the most important staining procedure in the bacteriology laboratory because it differentiates between gram-positive and gram-negative bacteria. Another one is the acid-fast stain, which is specifically used to identify Mycobacterium species. In the first step, crystal violet is added to the cells in a smear. It stains the malt the same purple color. The second and key differentiating step is the addition of the mordant or the gram-S iodine. Application of alcohol in the third step dissolves lipids of the outer membrane and removes the dye from the peptidoglycan layer of the gram-negative cells. Because gram-negative bacteria are colorless after decolorization, their presence is demonstrated by applying the counter-stain saffron in the final step. The different results in the gram stain are due to the differences in the structure of the cell wall and how it reacts to the series of reagents applied to the cells. Therefore, if a bacterium is blue to purple at the end of the gram staining procedure, it is said to be gram-positive. If on the other hand, it ends up being pink to red, it is said to be gram-negative. Now take a look at the characteristics of some important gram-positive pathogenic bacteria. Take note of their morphology as well as the diseases it causes. Then here are some of the gram-negative pathogenic bacteria. This time let's recall the steps in acid-fast staining. In this procedure, carbofuscin is used first to flood the smear of bacterial cell and then using heat as a mordant either by heating equipment or holding a Bunsen burner flame under the slide until it stinks. The heat softens the waxes enabling the sting to penetrate. Next, a decolorizing agent, usually a mixture of acid and alcohol, is then used in an attempt to remove the red color from the cells. Finally, a counterstain using methylene blue is applied. Because mycobacteria are not decolorized by the acid-alcohol mixture, they are said to be acid-fast. Most other bacteria that are decolorized by the acid-alcohol treatment, thus retaining the blue color of the secondary stain, are non-acid-fast. This acid-fast stain is especially useful in the tuberculosis laboratory or TB lab, where the acid-fast mycobacteria are readily seen as red bacilli against a blue or green background in a sputum specimen from a tuberculosis patient. Rickettsias, chlamydias, and mycoplasmas are bacteria, but they do not possess all the attributes of typical bacterial cells. Thus, they are often referred to as unique bacteria because they are so small and difficult to isolate. They were formerly thought to be viruses. Rickettsias are obligate intracellular pathogens that cause diseases in humans and other animals. As the name implies, an obligate intracellular pathogen is a pathogen that must live within a host cell. It should also be noted that these organisms have no connection to the disease called rickets, which is the result of vitamin D deficiency. Rickettsias have a gram-negative cell wall. All diseases caused by Rickettsia species are arthropod-borne, meaning that they are transmitted by arthropod vectors or carriers such as slice fish and ticks by their bites or waste products. So medically important bacteria that are closely related to Rickettsias are Cachiella burnettii, Bartonella quintana, Ehrlichia species, and Anaplasma species. The term chlamydias refers to chlamydia species and closely related organisms. Chlamydias are referred to as energy parasites. Although they can produce adenosine triphosphate molecules, they preferentially use ATP molecules produced by their host cells. Unlike Rickettsias, they transmit the disease by arthropods. Chlamydias are transferred by inhalation of aerosols or by direct contact between hosts. Medically important chlamydias are Chlamydia trachomatis, which cause different diseases, including trachoma, which is the leading cause of blindness in the world. We also have Chamaedophila pneumoniae and Chamaedophila psittaci. And then we have mycoplasmas, which are the smallest of the cellular microbes. Because they lack cell walls, they assume many shapes from cocoa to filamentos. Thus they appear pleomorphic when examined microscopically. Sometimes they are confused with CWD forms of bacteria, which were described earlier. However, even in the most favorable growth media, mycoplasmas are not able to produce cell walls, which is not true for CWD bacteria. Therefore, do not confuse mycoplasma with mycobacterium because each is a genus of bacteria. The unique thing about mycoplasma species is that they lack cell walls, thus are resistant to treatment with penicillin and other antibiotics that work by inhibiting cell wall synthesis. Pathogenic mycoplasmas are Mycoplasma pneumoniae in Mycoplasma genitalum, which cause primary atypical pneumonia in genitourinary infections, respectively. It was believed that bacteria and archaea diverge from a common ancestor relatively soon after life began on this planet. They were once referred to as archaebacteria. Archaea, which means ancient, was originally assigned when it was thought that these prokaryotes evolved earlier than bacteria. Now there is considerable debate as to whether bacteria or archaea came first genetically. Even though they are prokaryotes, archaea are more closely related to eukaryotes than they are to bacteria. Archaea vary widely in shape, some are coca, some are bacilli, and others form long filaments. Many, but not all archaea are extremophiles in the sense that they live in extreme environments such as extremely acidic, alkaline, hot, cold or salty environments or environments where there is extremely high pressure. It's time to conduct her own study on each of the bacterium that we have discussed in this laboratory exercise. You will be classifying the bacteria based on its gram staining results, morphology, and associated diseases. You will also need your creative hands in drawing some of those medically important bacteria. Test what you have learned about viruses and bacteria. This quiz will be open until Friday, so make sure to review your notes before taking the quiz.